Opioids are one of the most controversial drugs of modern times. This class of extreme pain relieving drug is a major public issue, but how exactly do they work? There are two types of opioids, endogenous and exogenous. Endogenous opioids such as endorphin and dynorphin are produced by the body that act on opioid receptors to the central nervous system as a natural response to pain and other stimuli. These opioids are not harmful to the human body since the brain controls the amount that is released. On the other hand, exogenous opioids are artificially made, which can be harmful depending on the amount that is ingested. As exogenous opioids enter the bloodstream, they bind to one of the three types of G protein-coupled receptors, mu, kappa, and delta, on the cell membrane. G protein CRs are receptors that, when activated by GPCR ligands or monoamines, activate G proteins that are bounded to them. When a ligand binds to the GPCR, it causes a conformational change in the GPCR, which allows it to act as a guanine nucleotide exchange factor. Then it can activate an associated G protein by exchanging the GDP bound to the G protein for a GTP, similar to ATP. These proteins then dissociate from the receptor and perform various functions in the cell. In the case of opioids, the alpha complex and the B complex dissociate together and trigger the inhibition of adenylyl cyclase, an enzyme which converts ATP into the secondary cellular signaling molecule cyclic adenosine monophosphate, or CAM. These G proteins also inhibit opioid receptor signal transduction. This relates to the ability to regulate calcium ion transport into the membrane and boost potassium ion transport out. Channel deactivation occurs after GTP to GDP hydrolysis and with the removal of G beta gamma. This process causes cellular hyperpolarization and inhibits tonic neural activity. In many cases, mediated interactions with this inward potassium channel were seen to greatly affect neural excitability.
In nerve cells, this polarization of the cells lowers amounts of signaling molecules and translates to the neuronal transmitted activity decreasing. It also triggers release of dopamine in the brain, creating a high, and breathing slows. This pathway is effective in reducing pain sensors in the body, therefore leading to a high or above average state of feeling. The reward pathway, on the contrary, can exponentially increase the potential chance of drug abuse, because consuming opioids is highly pleasurable to the brain. In an opioid overdose, excess opioids bind to their GPCRs and trigger a major decrease in neuronal transmittance, to the point that breathing slows to a very slow rate, and may even cease. What can we medically do about this? Common side effects of opioid administration include sedation, dizziness, nausea, vomiting, constipation, physical dependence, tolerance, and respiratory depression. Physical dependence and addiction are clinical concerns that may prevent proper prescribing and in turn inadequate pain management. Less common side effects may include delayed gastric emptying, hyperaglasia, immunologic and hormonal dysfunction, muscle rigidity, and spasms. Luckily, naloxone is a drug that competitively inhibits opioids. Naloxone has a similar shape and structure to opioids, so when flooded into an overdose system, can bind to opioid receptors at a high rate, preventing opioid binding. Therefore, naloxone essentially reverses the negative effects of opioid usage. Today, it is important we educate all on the workings, dangers, and treatments of opioids.